George Paul, for those of you who don't know, is a master's student. Actually, he's a recently graduated master's student. He defended his thesis, but we found out that he still required a seminar, and I think he was more than pleased to give you a seminar here today about his work. George Paul is one of the ones that I consider to be a kind of a success story. He came to my lab as an honors student, so through your honors program, and some of you might know that the honors program offers a 4-in-1, which means that you can do your honors program, and then if you want to, you can extend it by 1 year and convert your honors work to a master's degree, which is what George Paul opted to do. He came to talk to me about it. I was very pleased that he opted to do that. Everything that he is presenting you here today, I would like to take credit for, but I cannot, because although George Paul probably learned a lot through his master's project and thesis, I learned more because this is all entirely new to me. So I learned from George Paul, and I take that as being a win-win situation. So George Paul, if you don't mind telling us about some of your work, and then I'm sure there's time for some questions afterwards. Thanks, George Paul.
Sounds good. Thanks for the introduction. So if we're ready to start, I guess I'll get going here. So I'm going to talk to you guys today about the gut microbiome in the context of autism spectrum disorder, which is essentially what my thesis kind of investigated. So as a little bit of preamble, I'm going to tell you about autism spectrum disorder. Basically, it's a psychiatric disorder. It's changed a couple of times throughout the different iterations of the DSM, but it's generally thought to be a neurodevelopmental disorder, which occurs sometime during early childhood. The symptoms that you tend to see and what it's diagnosed on the basis of, the most prominent one would be impaired communication, impaired difficulties with social interaction. You can see sensory processing issues like hyper or hyposensitivities to different stimuli, things like that. And overall, these things together can impact someone's quality of life quite a lot, but to varying degrees. The symptoms kind of show up differently in everyone, which is how you get the spectrum in autism spectrum disorder. So the current kind of literature around autism considers it as more than just a brain disorder. We tend to see differences in sort of immune system changes going on. For one, we'll see neuroinflammation in people with autism, and the degree of that inflammation can be correlated with symptom severity. We also see changes in the functioning of the immune system, such that children with ASD tend to experience more viral infections than children without. So there seems to be kind of functional parameters changing there. But some of the more modern stuff and what this thesis focused on was differences in gut microbiome composition. So I'm going to highlight two kind of interesting papers for you guys here. First one was a study in which they gave a bunch of children with regressive onset autism oral vancomycin, which is a broad spectrum antibiotic. And the gut microbiome, all those little microbes that live in your intestinal tract, if you take a broad spectrum antibiotic, you're basically nuking their home. So everything dies off. What they found in these children, huge improvement in their symptoms, like sometimes miraculously so. And this improvement kind of went away as they stopped taking the antibiotics. So this generated a whole bunch of interest, and ensuing research has shown that you can take a fecal microbiota transplant from people with ASD, put it in mice, and reliably induce behavioral symptoms of the disorder. So that's very interesting. It kind of blows the whole autism is just a brain disorder thing out of the water. And before I get back into that, I'm going to pivot for a second and just explain a little bit about the mouse model of autism that we used in this study. So it involved something called APP. So APP is a self-surface protein. It's expressed all over your neurons. I think it has something to do with cell adhesion and a couple of signaling functions. But there's an enzyme called alpha secretase, which can cleave this protein into a soluble form called solid precursor protein alpha, so SAPP alpha. What is, you can think of it as one of the good guys. You give it in the good guys. It's neurotrophic. It increases myelination. It facilitates the branching of dendrites, and it basically opposes apoptotic signaling in neurons. What happens when you have too much of this? Well, all those things still occur, but at an exaggerated level. And why is that a bad thing? Well, during the first couple of years of your life, when your brain is very rapidly developing, you're basically forming tons and tons of synapses every second. I think I read somewhere that it's like 1 million new synapses every second within the first 5 years of life. Not all of those synapses are useful, not all of them are appropriate, and many of them, it's actually a bad thing to have them stick around. So you have this constant kind of balance of apoptotic signaling and pro-growth signaling to kind of make sure that those aberrant or inactive pathways are being pruned away, and you don't get all these connections between parts of the brain that shouldn't normally talk. If you have too much SAPP alpha floating around, that pruning doesn't occur properly, and that's thought to be what's happening in autism and what's associated with the symptoms that are occurring. So I'm just going to show you a couple of studies here which kind of add some evidence for this hypothesis. One is that plasma levels of SAPP alpha tend to be increased in patients with autism, particularly severe autism. If you take the brains of people with ASD and you look through them, you will find increases in the alpha secretase enzyme in general, which is again the thing that cleaves APP into SAPP alpha. And a couple down in Florida, a number of researchers created this mouse model, a transgenic mouse model, which overexpresses SAPP alpha in a neuronal promoter gene. So what happens when you overexpress SAPP alpha in these mice, you get autism-like behavioral, neurological, and immunological phenotypes. So that's very interesting. And then my fourth point there is just to call back to that other slide. We know that ASD is associated with significant differences in gut microbiome composition. Does the same happen in the SAPP alpha mouse model? That is my research question. So we know that the SAPP alpha model correlates with autism in humans on a variety of different points. Does it correlate in this particular point as well? So my hypothesis was that SAPP alpha overexpression would alter the composition of the gut microbiome. So how did I test this? Well, I had to use a method called amplicon sequencing. And that's where my other supervisor, Dr. Janet Hill, comes in. She basically taught me all about this. The gene, the amplicon that I use to do this is a thing called CPN60. So CPN60 is a gene. It codes for chaperonin 60, which is a heat shock protein. Basically, all bacteria need it to be a functioning cell. You can't get away with it. So, excuse me, all the bacteria that have this gene, they have particular functional regions that have to be conserved. Those are the parts that you need to have the heat shock function, right? But then there's also a region in the middle of it, the variable region, which is essentially unique between different bacteria. Sorry, my phone's buzzing here. So this variable region, it's so unique between different bacteria, you can actually use it as sort of an identifier, almost like a barcode. So Dr. Hill and her colleagues, they designed CPN60 primers. So these primers bind to the conserved regions on either end of that variable region in the middle there. And when you amplify them, you basically get a transcription of this barcode, this bacterial barcode. And Dr. Hill maintains this database of barcodes and the species of bacteria that they correlate with. So you get a whole list of barcodes, you plug it into this database, and it spits out a list of the species of bacteria that you have in a particular sample. So let's just get into the study design here. I had mice aged 3 months of age, 3-month-old mice kind of simulated the childhood condition in humans, which is when autism is most commonly diagnosed. We had a total N of 28. We took fecal samples from these mice. So you take the fecal sample, you extract all the DNA from it, and contained in that DNA is all the DNA of all the bacteria that essentially reside in that GI tract that kind of take a ride out with the fecal sample. So you extract the DNA, you purify it, you amplify it with the CPN60 primers. You get an amplicon sequencing library, which you throw in this machine called an Illumina MySeq. It gives you a ton of data, gigabytes upon gigabytes, which you then have to process. And this is where the kind of bioinformatics stuff comes into it. We do a whole bunch of quality control and data processing, and we're essentially left with that list of barcodes, as I mentioned before. So taking that list of barcodes, we plug it into Dr. Hill's database, we get a list of species, all the different species of bacteria that show up in each sample and how many times each of them show up. So once you have that information, you could do your visualization, your statistics, all sorts of fun stuff. Again, I had to learn a ton of like bioinformatics and coding, and I'm not the most computer literate, so this is hard. I won't go into it unless you guys have more questions. But that'll lead us right into our results. So what did I find from doing all this stuff? First of all, we're going to look at overall composition. So one of the ways of looking at the overall composition of a community is alpha diversity. So I used two measures of alpha diversity. The first one was Faith's phylogenetic diversity, you can think of as richness, and that's basically asking how many different species are in this community. So if I look at one forest and I go, how many different trees are here? There's 10 different species of tree. And if I look at another forest, oh, there's only 2 different species of tree. So forest number 1 is more diverse than forest number 2. These graphs on the right here essentially show that. So Faith's phylogenetic diversity, each one of these numbers represents a sample here. When I looked at this statistically, there weren't really any significant differences between any of these samples. So that's the different sample groups. I segregated them in terms of genotype and sex. So we got wild type males, heterozygous males, wild type females, heterozygous females. No differences in terms of richness. Same thing goes for evenness. So evenness is another alpha diversity measure. It's like looking at those same forests and actually considering the number of each species that are present. So forest number 1, 10 species. How many trees are in that forest? Maybe there's 10 trees. If you look at forest number 2, there's 2 different species of tree, but there's 10,000 trees of the first species and like 10 of the second species. That's going to have implications for your community and the ecology that's going on. Either way, no significant differences. Same kind of story with evenness. So I looked at another measure of overall diversity after doing that. This is kind of the flip side of that question. Beta diversity is comparing 2 communities directly. It's going, which species do these communities have in common, and how many of those species are present, and where are they similar, and where are they dissimilar? And if you look at the bottom right of this graph here, this is a thing called a PCA plot. We can see the different colors of each sample group are represented here. The important thing is that all of these ellipses, they all intersect. So there's no real differences between these groups. Each one of these little dots here represents one sample. There's no real skewing. Like if you saw a bunch of them in the corner up here, you could say, oh, that's that distinct group, but not much difference going on. So both of these measurements, alpha and beta diversity, they are concerned with the big picture, like the huge broad scale community-wide survey type of thing. There's another way that you can look at this question, which is individually. You go through the different species and you go, does this species show up in this community or does it not? So that was the next thing I did. That's kind of the final point of investigation here. Differential abundance of individual features. So there's a program called ALDEX2 that does a lot of this for you. I plugged my different groups in there and compared them. I found a bunch of differences between groups. I'm not going to go into all of them. I'm going to focus on one in particular because the CPN database, it has kind of a standard for identification, which is that the sequence that shows up, it has to be at least an 85% match to the reference sequence in the database for me to say, okay, I've identified that, that's what that is. Anything below 85%, we can't really say for sure, and we'd have to do a bunch of other testing to figure out who that bacteria is. But out of these differences I found, this is the only one that met that criteria and actually blew it away. It was like 100% match. So Akkermansia muciniphila, I can say for pretty sure, it showed up less in the transgenic males than in the wild type males. It showed up less in the transgenic females than in the wild type females. So across the board, the transgenic condition is associated with less of this bacteria than the normal condition. And then if I segregated by sex, if I compared control males and control females, we saw less of this bacteria in females in general. But then when I compared the transgenics, there was no difference at all. So there seems to be a sex difference at baseline, which doesn't persist into the transgenic condition, which is weird, but interesting. So I did a bit of research into this particular bacteria, And it turns out that there's a bunch of connections between Akkermansia muciniphila and autism. If you look up reduced abundance of Akkermansia muciniphila, you'll find it's been associated with a variety of different illnesses, obesity, diabetes, things like ulcerative colitis, inflammatory bowel disease, and also in children with IgE-related atopic illnesses like allergies, like asthma, things like that. So there's a lot of theory that this bacteria influences or aids host immune function in some way, and can alter your metabolism. ASD is frequently associated with all of these things. They're all complications that occur more often in ASD than in neurotypical children. And at least one study has shown that this bacteria shows up in significantly reduced proportions in children with autism, and then compared to their siblings, and then in their siblings compared to non-sibling controls. So there seems to be a genetic or heritable component that influences either autism or your likelihood of having less of this bacteria. There's some sort of connection going on there. We also see, and this is quite interesting, is that if you have decreases in this proportional abundance of Akkermansia muciniphila, proportional abundance meaning less Akkermansia compared to less of everything else that's in there, that's correlated with heightened anxiety and depressive behavior in rodents in response to social stress. So that kind of results in impaired sociability. Rodents are very social creatures by nature. What you get here is a phenotype with less social interaction. So that kind of correlates with autism in humans. So yeah, Akkermansia muciniphila, very interesting bacteria, worth looking up, all sorts of implications on host metabolism. And it can actually influence host gene expression directly. So it eats the mucus in our intestines, which is kind of why it got its name, Akkermansia muciniphila, lover of mucus. It can actually influence the intestinal cells to produce more mucus so that it can ensure it has a good growth environment and it can keep eating. So it's kind of like your master, maybe. But that ties into this whole idea of the gut-brain axis. And that actually, bringing back to this mouse model, what I've shown here actually, it improves the status of this mouse model. It tells us that this is actually a very good mouse model because we know that SAPP alpha is associated with immunological differences in these mice. We know that, the gene expression, it's a transgenic mouse. We know that that's going on here. We can show with this study that SAPP alpha influences the gut microbiome composition, and that all is thought to tie into the altered brain development that's going on in autism spectrum disorder and the behavioral phenotypes. So now that we know that this is a really good mouse model, we've kind of tied together the SAPP alpha side of human autism with the gut microbiome side. There's a whole bunch of other questions that we can actually address using this model. So some of the big ones, the more interesting ones, how does age affect this? Because we used one time cohort of mice that were all 3 months of age. If they got older, how would this change? How does sex affect this? Does sex, excuse me, there seems to be a sex difference at baseline, but not one in the transgenic group. Why? Why isn't there? And why is there one at baseline? That's a question that could be asked. And then how does diet influence this? Because there's a common trend among the parents of children with autism to try all these kind of crash diets, like, the ketogenic diet will cure your child's autism, or gluten-free diets, or casein-free, all these things. How does diet influence the microbiome in this context? And does it influence the amount of Akkermansia muciniphila there? And would it change in these mice because they were all eating the same kind of food? So some of the more kind of practical questions that definitely do need to be answered. Can the effects that I showed in the study on Akkermansia muciniphila abundance be replicated in a higher powered study with more mice? And as well, is this SAPP alpha overexpression associated with evidence of GI pathology or inflammation? Because we know that reduced abundance of Akkermansia shows up in ulcerative colitis and IBD. And those things are very common in people with autism spectrum disorder. So does this cause changes to the intestinal tract pathologically in the mice? Do we see, or changes in mucin production? That's another good question. And I guess that's the end of it. So I'd like to acknowledge the following individuals. So Dr. Mousseau and Dr. Hill for being my supervisors. my committee members, Dr. Widdenmeyer and Dr. Howland, the members of the Hill Lab, Shampika, who taught me how to do all that magical stuff with the mouse poop, and then Scott, who put up with me constantly blowing up his phone to ask him all these questions about the bioinformatics. And then the members of the cell signaling lab, of course, they were the ones who helped me collect the poop in the first place, which was a fun job. Prairie Diagnostic Services, and the LASU staff for taking care of the mice. So these are my references here, and I guess if you guys have any questions, you can go for it.
Thanks, George Paul. So are there any questions around the table? And I guess what I'll do is I'll ask the students first to see if they have any. And if the students then run out of questions, then maybe we can step into faculty.
Yeah, actually, I have a question for you, GP. First of all, that was a fantastic talk. Thank you so much. My question is regarding the vancomycin use in the people with ASD. And I was wondering if you knew if there were any long-term effects of taking the vancomycin in people with ASD compared to people without ASD. Mm-hmm. Like it chronically, as in, you know, longer than what a recommended dose would be from a pharmacist, like over a period of months or years.
As far as I know, I've only read that one study, and I don't know that there have been any studies showing like chronic long-term use. I know that in this study, the changes went away when you stopped. But the thing is that people who are on chronic antibiotics for other things, that does have a huge impact on your gut microbiome, and not always in a good way. And it can make you very susceptible to things like C. difficile or things like that, you know, like hospital-borne illnesses, things like that. So I think that they probably wouldn't recommend that, given that you can build resistances in these bacteria, and then you get, you know, in the general population, you have all these resistant bacteria lying around, but I don't know that's ever been studied. I know that fecal microbiota transplants have been studied in people with autism, and there seem to be promising results so far. It's still kind of ongoing. But yeah, thanks for that question.
Yeah, thanks.
Very good question. Thanks, Kirk. Are there other questions around the table? If not, maybe then what I'll do is I'll reach out to faculty. I see that Julia, Dr. Bugner might have a question. So please, Julia, go ahead.
Yeah, thanks very much. And George Paul, that was a fantastic talk. It was really, really interesting. And you mentioned how bacteria are our masters. And I know you said that in kind of a facetious way, but I've also read that gut flora can influence what we decide to eat without us realizing that's what's going on. So I was wondering, in reference to the comment that you made, that is true, I believe, that often kids on the spectrum, as well as maybe kids in general, have particular food preferences that can be very strong. Whether there's anything known about these food preferences either being led by the gut flora, or whether the food preferences are to try and correct for a typical gut flora. What do you think?
I don't know. I think that's a very good question. And that's one of those things that if you could tie those things together, I mean, it's waiting for the right kind of study, and you'd answer some questions, particular change. I have seen a lot of things about that kind of food, like very specific food preferences. Like I worked with a kid who he'd only eat grilled cheese, and his parents they were at their wits' end, but it was like he had one specific, he couldn't eat other foods. And I don't know, the thing is that when you have very specified or restricted food or a dietary kind of habits, that will change your microbiome as well. So it could be that the microbiome changes are the result of this, or that they're precipitating it, and it's very much unknown territory right now. But yeah, thank you for that question.
Yeah, and I suppose, and maybe it's already been done, that you know, some kids on the spectrum have been given oral doses of gut bacteria to try and correct for it. I suppose that has been tried. Do you know?
Yeah, it's been tried. There's a couple of different probiotics that have been tried. There's a problem with just giving like one particular species though, because you have thousands of species in your microbiome, and you don't know that this one is going to, maybe there's a whole chain of interactions going on, and you might need 3 or 4 different bacteria or different strains. So there's a lot of ongoing studies right now which are looking into this. Like, it is very recent stuff, and I'm guessing that what I'm saying to you is probably going to be outdated by the time it leaves my mouth. So yeah.
That's great. Thank you very much. I'll stop monopolizing your question period and offer the mic to someone else. Thank you.
No worries. Thank you.
Hi, George Paul. Can I ask a question?
Sure, go ahead, Lane. Go. Yeah.
You mentioned that SAPP alpha and alpha secretase were upregulated in these ASD children. Do you know if there's any changes in serotonin levels? Because I seem to recall that serotonin through 5-HT4 or something can directly affect expression of alpha secretase.
Yeah. Autism, it's actually one of the most, it's actually, they reported increases in this chemical, this 5-HT, whatever it is, before they'd actually, in children with autism, before they'd actually classified serotonin as a neurotransmitter. So they're going, there's elevations in this, whatever it is, and then they discovered serotonin like 20 years later, or they characterized it. So it's very commonly known that autism is associated with hyper serotonemia, I guess you would say. And that is a good point, that 5-HT4R receptors do increase the expression of SAPP alpha, and they're found in the gut. They're found all over the gut, and they're actually used, I'm guessing you know this, in drugs that I think target constipation and things like that, because they're linked with GI motility. But if you have more serotonin than normal being in your bloodstream, and it's able to get to these receptors and increase the expression of SAPP alpha, there's a mechanism right there for why you might have overexpression in these children. It's very hard to tell which comes first, though, like where it starts. But that's a great
to a second question.
Sure.
I don't know anything about ASD, so are they treated with any of these antidepressants or anything like that to sort of try and alter serotonin levels? And are they effective?
It's very common to see kind of polypharmacy going on. You'll see antipsychotics or antidepressants, things like this, but there's no uniform thing of, oh, we put these kids on SSRIs and they get better. There's no, as far as I know, it's all a symptom by symptom individual basis. So I don't know that it's as simple as just, oh, we can block serotonin, we can do something and improvement in symptoms. It's very much like, oh, this kid has very bad anxious symptoms, we can try and give them an SSRI. But yeah, that's as far as I can talk on that. I'm sorry.
Okay, good. Thank you. Thank you very much. Excellent talk.
Thanks for the questions.
I think we're probably running pretty close on time, but just as a follow-up to your question there, Lane, I'm sure this is something you know, or maybe you don't know, that the gut produces between 95 to 97% of all the body's 5-HT. So the fact that there's a gut dysbiosis in these children and that there's a downstream serotonergic effect is not a surprise, because they're just saturated with 5-HT. And of course, 5-HT in the blood makes its way over into the brain through specific transporters. And as a follow-up to Julia's question, just something maybe George Paul wasn't aware about, because there was some other work done with these mice previously in our lab, is that there's a difference in the ability of male and female transgenic mice, these SAPP alpha mice, to smell. So we tested them in terms of their odor function, and the females have got a significant loss of odor function, transgene, versus the male that don't seem to get bothered by the expression of this transgene in terms of odor, whatever the proper term is. I'm sure there's some term out there, but anyway. So thank you very much. I think are there any other questions around the table? Students, maybe a couple of the questions from the faculty triggered some more. Thanks, Dan. If not, then I will thank you very much, George Paul. Very nicely done, and I'll give the floor back to Dr. Eames.